Okay. So hi, everybody. Thanks for coming out on what feels like summer in San Diego in March. A little change from the monotonously beautiful weather that San Diego normally has. I am here. I'm Todd Butler. I am Dean of the College of Arts and Letters. This is already guaranteed to be the best meeting I have all day, in part because we get to talk about interesting things. And this is part of the College of Arts and Letters has had a running lecture series called HumeTech over the past year, which is investigating and opening up spaces to talk about the intersection between humanity and technology. Upcoming things, in fact, this is part of our what we're calling Creativity Week, for reasons that I think will be clear in terms of what we're going to talk about today. Tomorrow is going to be talking about electronic poetry. Later in the next month, in April, we'll be having a series on the future of work, broadly construed. We'll be having a law professor from UC Irvine talk about algorithmic wage discrimination. That is what happens when algorithms can sort out how much your Uber driver is paid or your DoorDash driver, et cetera. We'll also have a panel at that point coming up on the future of war, which we scheduled before, the future arrived. Right now, what does that mean for technology and what we see in Ukraine and elsewhere in terms of the advances in technology to intersect with the human experience of war? We'll also be having a session on, particularly for undergraduates, on career prospects and the growing marketplace for responsible AI. That's in part linked to SDSU's first undergraduate degree in artificial intelligence, which happens to live in the College of Arts and Letters. It's a BS in artificial intelligence and human responsibility. Half computational linguistics, visual pattern recognition, data management, et cetera, and then half politics, public policy, ethics, and environmental impact. It is new this year. We took it from ideation to submission in 3 weeks last year, and then it took a year to get through the CSU. But we'd like to still say that academics can move fast and make things. And making things actually is what we're here to talk about today. I'm here joined with Brian He. Is that, did I say
yeah,
right?
that's
correct.
Awesome.
Good. Brian is an assistant professor of chemical engineering at Stanford. He's also the Dieter Schwartz Fellow, the Dieter Schwartz Foundation Stanford Data Science Faculty Fellow, and an innovation investigator at the ARC Institute, where he supervises the Laboratory of Evolutionary Design. He was previously a Stanford Science Fellow at Stanford University School of Medicine, a researcher at Meta AI, and was trained at CSAIL, the Computer Science and Artificial Intelligence Lab at MIT. He is also a proud San Diegan, born and raised, graduated from Scripps Ranch.
Go Falcons, yeah.
Right, exactly. Had his graduation on this campus as well, and we're really super excited to welcome him back. Brian, thanks for coming back home.
Thank you for having me. I'm excited.
Yeah. So let me give you a little sense. Brian, why don't you, just as a start for folks here to get into, I've already charted a bit of a circuitous path for you from Scripps Ranch to Stanford and back, dropping into Meta, going up to MIT. You're now a chemical engineer, which is strange when we've got a whole bunch of biologists here, et cetera. Many of the challenging questions that we investigate, of course, are multi-entry point questions. So walk us through how you got to where you are right now.
Yeah, it is somewhat a circuitous path, and I think it reflects how interdisciplinary modern science is. Even if you want to make progress, you need to have capabilities in computer science, in biology, in chemistry. So in terms of I think you had a great summary. After graduating in the stadium here in San Diego, I then did my undergraduate at Stanford as well. So I kind of never left. I did an undergraduate degree in computer science, where I actually specialized a lot in computer systems, operating systems, compilers, distributed systems, which ended up being very useful for AI, which is now trained on large clusters, and it's a huge distributed training algorithm, for example, to make the large language models. I then did a PhD at MIT, where I also did computer science, but then was wanting to pivot more into machine learning at the time. The AI was very new. It was almost like AI had gone through various winters, and it was almost like bad to be in AI because there's so much stigma attached to it. You would say I'm in machine learning, and then I ended up joining a lab applying machine learning to problems in biology. Because I've always been a pretty practical person, I like the intellectual challenge. It seemed at the time that biology was very, very hard to solve with any kind of computational tools. And I also like the idea that maybe one day the algorithms that I was developing would be useful to patients' lives and would be deployed as drugs or therapeutics. So I joined a lab doing machine learning in biology. And then towards the end of this, I started training these very, very small language models, like million parameters on thousands of sequences, on protein sequences, and then basically continued that line of work through. Then I was a Stanford Science Fellow at Stanford, where I retrained as a protein biochemist because I felt like I was making all these predictions in biology and not being able to test them experimentally. Simultaneously, I also joined Facebook AI research, or Meta AI as it's called now. And there I was working on a team that was also training language models on proteins, but at much larger scale, at the scale of, at an industrial scale with lots of GPUs. I felt that was a really interesting time. I was running experiments in the lab, and while waiting for the experiments to complete, I was launching neural network training jobs on a huge cluster and waiting for those to complete as well. And then I bring all of these techniques into my lab now, which kind of combines and various methodologies spanning AI, spanning protein biochemistry, but now we're also doing things in genomics, and also that systems knowledge, that distributed systems knowledge, is actually finally becoming useful again because we train large models at scale. So
Sure.
that is a brief summary of the path.
Well, and so one of the things that I hear out of that is the notion that our careers don't stay the same. Who we are never stays the same. That we evolve as scientists, as researchers, as individuals, and where we are today may not be where we are 5 or 10 years ago, both as a field but as individual researchers. So let me bring our audience up to date a little bit on what brought Brian to my attention and why we brought him down here. And so it's about your group, the EVO group. And you recently demonstrated that a generative AI model trained on massive genomic data sets could design not just proteins, which is something that we've accomplished before, but design entire viral genomes. And some of those designs, some of those viral genomes actually produced functioning bacteriophages capable of killing bacteria. Okay, now for some people that will sound like science fiction. Some people that will sound very exciting. A few people will see how it's written up in the Washington Post as Brian, his group just created life, and immediately click on the clickbait title. That would be me, and that. But so tell me, actually, beneath all that description, beneath that hype, what exactly did your group do?
I mean, that was a great summary. And yeah, I think that the Daily Mail headline was even more clickbaity than the Washington Post.
What was it?
It was like AI apocalypse or something like that. I'm definitely not intending or even close to bringing any kind of AI apocalypse. The general contours of what you described are accurate. So the models that we trained are similar to the models that underlie ChatGPT. And these are models that are very large neural networks, and they're actually trained with a very simple objective. So they're trained to predict the value of the next word in a sequence. Actually, I have a... Okay, so this is the next token prediction task, right? And as someone who's read Shakespeare, you can probably predict the value of the next word in that first sentence. We actually train models with the same objective, but on DNA sequences. And the idea is that in language, for example, even though you're predicting the next word in a sequence, this task of next word prediction actually encodes many complex tasks within it. So you not only teach the model aspects of grammar, but you also can potentially teach the model things about reasoning or even mathematics. And the idea behind these large language models for DNA is that you can also teach the model not just things about individual base pairs, but also higher order patterns of how biology has put functional genomes together. And the models are generative models as well. So in the same way that you can ask ChatGPT to write code for you or write an email for you, these models with actually the same underlying algorithms can write new DNA sequences for you. And one very key idea behind training a model on DNA is that DNA is this fundamental information code that all of life basically needs to store its information in. And so if you train a model on DNA sequences, in theory, you can generate just DNA sequences that you want. And because DNA is this universal code in biology, potentially the things you can design in DNA are quite complex and quite interesting. For example, the human genome is fully composed of DNA, but also the genomes of bacteriophages or simple viruses are encoded in DNA or RNA. So we wanted to demonstrate that these models were not just capable of generating individual proteins. As you probably know, methods for generating proteins with AI won the Nobel Prize 2 years ago in chemistry. It was shared with the developers of AlphaFold. So we think that AI can definitely generate proteins, and proteins are very interesting. But we also want to go beyond, the scale of individual proteins. So I know at the left is the scale of individual proteins, and on the right is the scale of what a typical cell would look like. And this is orders of magnitude more complicated than any individual protein. But in theory, all of this is encoded by DNA. So that brought us to wanting to generate these complete bacteriophage genomes, these viral genomes with the model.
Excellent. So you're looking at, so you have it here that HumeTech hosted the first talk by a future Nobel Prize winner.
No.
But let's actually. I'm curious. Let's push a little further. So you designed or you instruct and work with the LLM or generative AI model to generate possible genomes for bacteriophages.
Yeah.
And to give a little bit of insight for our audience, look like out of about 300 designs that you ran, about a small fraction, maybe 18, 16 actually
16,
worked.
Yeah.
What did those successes and failures teach you about the process of evolutionary design or life?
Yeah, it's actually quite interesting. So the way we test these designs is that the computer spits out just a text file of DNA, right? A, C, G, and T. But there's ways that we can then chemically synthesize the DNA. There's a company in Coralville, Iowa, for example, that will synthesize a bunch of DNA and you can ship it to any address. I think you can order some DNA if you wanted. Right now, we then can put those DNA sequences into bacteria and then see if the DNA sequences produce phages. And the way we know if the phages are working is because they kill the bacteria. So the liquid, if there's bacteria in it, has a lot of, it's very cloudy or turbid. If the bacteria start dying because the phages are real, then the liquid actually becomes clear. So we have this very simple way of seeing if the DNA actually encoded for functional phages. In total, we tested this library, it's called, or this collection of 300 phages, and we saw that 16 actually produced viable phages. So within that library of 300, we wanted to test different levels of novelty or diversity. So it did. So the success rate, so to speak, does go higher as you grow closer to nature. So there is some kind of. It's partly maybe those phages were just valid, but they didn't work in the particular system we used to test them. That could go into that, or the model could be somewhat struggling beyond some level of novelty to generate some valid phages. But of the phages that did work, especially within this kind of realm so there's various ways that people measure the novelty of a phage genome. So for example, at what's called the species level, people often say that's around 95% sequence identity to an existing phage. Around that level of, say, new species, the model is actually pretty good at generating functional genomes. At the level of what's like a new genus, that's like 70% identity to anything in nature, the model is still struggling. But we think we can, and that's a good point, we can learn from the failures to guide future rounds of model generations and potentially generate things that are more novel. So
Is the model struggling, or is it that you're uncovering some fundamental boundaries on what a phage actually is at some point, or that there are certain levels of novelty that just either push beyond what a phage is or what is sustainable in nature just by the very nature of what you're dealing with?
yeah, there's many aspects. I definitely think these models can teach us about phage biology and where the sequence evolutionary boundaries of what makes a valid phage, where those are. It also pushes the boundaries of our ability to test things in the lab. So if you just have some phage genomes, they could actually work in other bacteria, but we use a very specific safe strain of bacteria, E. coli C, in the lab. And so this limits the space of phages that would work in this particular strain of bacteria. And yes, there's probably some limitation in the underlying models themselves, because the one key assumption in machine learning is that you can train a model on existing data and then extrapolate from that data to produce new generations. And the models can generalize pretty well, but if they lack data in certain areas, if there's not a lot of data in some general space of phages, for example, the model won't be able to design things very well in that space.
So this gets into a question of providing that training data, who generates it, the human beings. And maybe let me ask you a broader question. Where does scientific intuition, human intuition, play a part even now in the process that you're using right now? What is the place for the scientist in this process in your lab?
Yes, there's an immense amount of human intuition and expertise that still goes on into designing things like a complete phage. So, for example, in this particular design process, the model spits out just raw DNA sequences. And I look at DNA sequences probably every day, and I still probably couldn't tell you the difference between a valid genome and something that was just a random scrambled DNA sequence that was completely randomly generated. We actually rely on computer algorithms to also then analyze that DNA and tell us if those genomes look reasonable. And there's a lot of human knowledge baked into that. The way we see it is it's almost like this is something that people do in nature. So you'll go out and sequence deep sea vents, or you'll go into the jungles of the Amazon and sequence DNA from these places. And then that will be one way you can discover new phage genomes or just new genomes in general. So that's one strategy. The way I kind of see these AI tools is it's an alternative strategy for generating new DNA sequences. So instead of having to go out into the Mariana Trench or something, or the Amazon to get new genetic diversity, or sequencing wastewater, an alternative strategy is to just sample from the generative model, sample from the AI model, and this gives you some diversity. And then we apply really the same set of tools to analyze the sequences, but rather than having those sequences come from nature, they come from the AI now. And then, so that is a big question. Are those tools that we have for uncovering and identifying the new biology also limited because they're based on very simple approaches developed in the 90s? So we are also interested in interpreting the language models themselves and seeing if they can actually be more sensitive to discovering new biology, both from nature or generated from an AI, in a way that the traditional tools are not. So that's a roundabout way of answering your question.
So where's the boundary between discovery and design?
Yeah.
That is, to what extent is the study of biology in your lab an engineering approach or a biology approach? And I'm going to grant that the distinction between discovery and design is perhaps artificial or necessarily binary, but I think it holds a certain kind of truth about an approach that someone has. So does biology become engineering truly in your lab?
It's engineering that we're getting more and more control. But as you said, right now, even the line between discovery and design does actually kind of get blurred, because we have these models. They're giant language models trained on giant databases that humans can't natively read the actual sequences. So like ACGT, I can't read in that language. So we have this kind of black box system which is the AI talking to another black box system, which is nature and evolution. And sometimes
Wait,
when
now let me just say, like the computer is talking to nature, and you're standing. I mean,
and we're like, yeah, we're kind of standing on the outside, and this is something that was not apparent to me until it happened. But there are cases in which the model will we use these models to generate proteins or new genomes all the time. And there's cases where actually the designs that the AI model has made will work in the lab according to some experiment. But we also have no idea biologically how that thing worked, other than, we know that it worked end to end. But like the mechanistic reason of why these proteins worked or why this phage genome worked, we still had to do a lot of hard traditional biochemistry and structural biology to find out why.
Okay.
So there's actually this kind of interesting possibility in which, rather than the AI models replacing humans or replacing traditional methodologies, the AI models could actually be generating designs that are just as interesting as things that we can discover from nature. Mm-hmm. And by studying the outputs of the AI systems, we can also learn an equal amount to studying from biological systems produced by natural evolution.
Great. And so I should say I'm going to make sure we reserve an ample time for questioning. So what I'm going to do is just seed some other topics as well so that we get at least some initial reactions. And I want to go back to something you said about a hope that you have for your work in the long term, that somehow that this makes an impact not solely on the field of biology or in large language models, but actually on patient outcomes.
Yeah.
And so talk a little bit about what in the long term you see as AI designed phages or this entire process mean for, let's say something like treating drug-resistant bacteria, which is a common and sustained and growing problem. That's a real problem, a solutions-oriented problem. How do we get from there to here? Or actually the here to there. Sorry about that.
We're excited, especially for this phage work. There's been this dream for a long time of using phages that are natural kind of predators of bacteria to also combat bacteria that causes infections at the hospital, for example. And part of the dream of phages as a way to replace or supplement traditional antibiotics is that the phages are much more specific. So rather than you taking some antibiotic and you completely nuking your gut microbiome, for example, you can actually just target the specific strain or species that's causing the infection. But also the phages can evolve alongside the natural bacteria. So if the bacteria acquires resistance to a traditional antibiotic like penicillin, you're kind of screwed, or you have to develop a new antibiotic. Well, the phage is this dynamic system too that will evolve with the pathogen. The problems with phage therapy are that it's also very difficult to match the phage to the bacteria to begin with. It's also hard to control the phages once you put them into a patient. And so, but we think that there's actually tremendous opportunity. In the paper, we showed that if you actually pool the AI-generated phages together, the 16 AI-generated phages, and combine them into what's called a cocktail, or like a mixture of phages, they can overcome resistance from on the bacterial side much better than either the just original version of the phage by itself, or even in subsequent work that we're going to disclose soon, even if you took phages from nature and pooled them together in a cocktail, the AI-generated phages actually do a lot better than just pooling natural phages together because they provide a lot more diversity. And so this is one way. We actually have this active collaboration with a doctor in the Stanford Hospital who deals with patients who suffer from lung infections that are caused by this bacteria called Pseudomonas aeruginosa. It's very, very bad. You don't want it. And we're actually interested in just taking some of their phages, adding additional AI-generated diversity, and maybe this cocktail of AI-generated phages is much better suited to overcoming these hospital-acquired infections because they're more resilient to evolution. So yeah, and more broadly, I think this is one of the clear benefits, I think, of acceleration in AI is the ability to actually make benefits on patients' lives and on drug discovery and on healthcare. I think this is one of the areas that are more clearly beneficial to humanity rather than generating deep fakes or something like that.
Yeah, I mean, I think that's a pretty low bar to clear in terms of that. But, so do you envision a future where scientists are routinely designing biological systems for medicines? I mean, you started with the idea of a biological cocktail, or sorry, an artificial cocktail that supersedes the efficacy of a natural mixture of bacteriophages and designing an artificial cocktail for that. So do you see a future where this is semi-standard practice for designing biological systems for medicine?
Oh, definitely. It's already becoming quite widespread in drug discovery for proteins and individual molecules. We have active collaborations. We have a collaboration with a biotech company on vaccine design and designing the proteins that go into vaccines. And these vaccines are going into monkeys soon and hopefully into people soon. There's also a small molecule drug that I helped develop as a graduate student, and it's finally also making its way into people. There's lots of pharma companies. Eli Lilly just announced a huge investment into AI. It's the pharma company that produces the GLP-1 peptide weight loss drugs. Basically all the pharma companies are making heavy investments in AI right now for individual molecules. Where I'm excited by is, in addition to the current drug discovery pipelines, is in developing new, more intelligent therapeutics that widen the net or widen the scope of diseases that we're able to overcome. Mm-hmm. So you mentioned designing entire systems or designing entire organisms. That is something we're actually very interested in revisiting. So there's been this long-standing dream in this field called synthetic biology of designing logic, like molecular logic, like AND gates or OR gates, or things that you would design in a computer, but with biological systems. And this is very important for many diseases, where one of the worst things you can do is like have something that kills all the cells, including the healthy cells, but not including the cancer cells, but also the healthy cells. We are interested in refining these systems for distinguishing what is a cancer cell, what is a healthy cell, with a lot of these generative tools. And for that, we'll need to design more complicated systems than an individual protein. We'll need to design logic involving multiple proteins and DNA elements and RNA elements too. But I think if we can make the therapeutics more intelligent, then this also expands the number of diseases that we're able to target with a drug or with some new therapy. So this is what we're actively working on right now.
Whether it's the black box approach that you talked about, or the implications of creating out new biological systems, I also want to introduce the challenges here, and I don't mean just technical challenges, the ethical challenges. Any number of bioethicists can also predict a future where this technology, this tool, this instrument, if you will, can enable ultimately not solely biological medicines that address rare diseases, but can enable the design of more harmful pathogens or toxins. And as I understand it, actually, even in your first design, you were taking steps to bound that principle by including and not including certain sets in the training data. Do you want to talk about that choice, how you thought through it as an ethical scientist and the implications thereof?
Yeah, one thing that we definitely take seriously are the safety, security, ethical implications of the research. And it is true that any new technology in biology, going back to when people were cloning DNA or editing DNA with CRISPR, can be used for a lot of good things. Like people with CRISPR, CRISPR is now curing very bad genetic diseases, sickle cell being one of them. But they can also be used for a lot of bad outcomes. And AI for biology is definitely another technological step change in biological capability that we do need to be very careful about. So the way we've been thinking about safety and security in these models is maybe there's definitely a multi-pronged approach. So the one thing we did for the EVO models that we wanted to open source the models freely for the community to use, but we also didn't want people to be able to accidentally generate viral sequences for viruses that infect humans, for example. So we actually excluded viral the sequences of viruses that can infect eukaryotes, which is this domain of life that includes humans, but also includes fungi, includes yeast. We excluded these sequences from the training data as just a precautionary measure.
Mm-hmm.
We also, for the particular phage work, made sure that everything we did was in non-pathogenic systems that we could test very easily. The phages are also constrained in their ability to infect by our algorithms. So we constrained their ability to infect cells to only bacterial cells. And the bacteria we used were also non-pathogenic. There's also, thankfully, a tremendous infrastructure in the biological community, especially going back to the Asilomar conference in the 1970s, for developing standards for biosafety. And we can actually, so we were in contact with Stanford's institutional review board, with various bioethicists at Stanford's main policy think tank, the Hoover Institution, which includes Drew Endy, who is a friend to people in the audience. So I think with all of these, we definitely wanted to be very thoughtful about how we were developing these models, making sure that at the very least we did not disseminate the ability to accidentally generate human viruses. I do think there is, though, there definitely is going to be some capability lift in the ability to engineer a bioweapon or a toxin. But in this regard, actually, I am fairly optimistic, because the same models that can be used to improve dangerous things can also be used just as well to detect dangerous things and to design countermeasures against dangerous things. And if you step back and think about which areas in biology actually benefit the most from AI tools, like the marginal benefit in various areas, actually, very unfortunately, it's not hard right now to design a new virus or a new pathogenic system. Nature does it all the time. For example, nature is actually very dangerous, and it's constantly producing very, very bad viruses that have pandemic potential, absent of any human intervention, right? The thing we can't do right now is defend against these threats. But where I see the AI models as being the most marginally beneficial is in helping us now acquire the capability to defend against threats from nature, or human-made threats as well. So overall, I think on the balance of things, as long as we advance these technologies safely and responsibly, then the marginal benefit to humanity actually very much sides with the people defending against threats and trying to cure disease. So this is why I'm optimistic about the technology.
So I'll have one more question, then I'll open it up so you all can be thinking about what you might want to ask. But I mean, that sounds familiar to a conversation I had with some folks at a national lab, where one of their priorities was identifying pathogenic problems or toxins they called risks beyond the list. That they could identify a whole list of possible categories of bioweapons or chemical weapons, but it was the ones that were novel in the way that they couldn't predict novel that really worried them, and that they were trying to figure out ways to address that. So that often gets the most press in terms of ethics. How about in terms of commercialization of these techniques, ownership of techniques? You gestured towards the ability of companies to invest and patent methodologies, forms of life or approach. Is that a concern ethically in terms of the work you're doing? And then how do we balance a certain kind of openness with the responsibility in the commercial realm as well as in the kind of danger realm of ethics?
Yeah, that's also an interesting dimension that we've considered a lot as well. In Silicon Valley, for example, there's a lot of pressure to commercialize and a big entrepreneurial culture, which I'm also very much open to. I think there are certain things in biology that require some amount of commercial infrastructure just to get to patients in general. But the way I see these models contribute more fundamentally, and they help researchers broadly answer questions in biology. They help researchers design new biology, but also we've been increasingly interested in using these models to discover new biology as well. And so we wanted to make these models as open as possible. This model is actually the largest fully open language model across any any domain, actually. So Evo2 is one of the models we use to generate the phages. And the model is actually what's called fully open. So we not only open source the model itself, but we open source the code for running the model, the code for training the model, and the data for training the model, which no one ever does. So and so we wanted to make sure that we're a bunch of researchers, we have a lot of philanthropic funding, and I want to make sure that these models return the maximal benefit to humanity. And we decided, in this case, that returning maximal benefit to humanity did look like just making the model fully open. I do think that navigating the clinical trials process and the regulatory process, doing all the work to get things through FDA approval, for example, this is probably best served maybe by a company or something, or a commercial entity, in a way that academia is not. And so there is some space, I think, for AI to also improve that process. Mm-hmm. And in the longer term, I definitely think that if you think that the AI tools make biology much more designable and much more engineerable and much more controllable, then that actually reduces, like, one of the problems, the reason why we spend $1 billion per drug is because there's a lot of infrastructure that goes into making sure the drugs are safe, that they actually work, and because there's like a 10% success rate once drugs make it into the clinic, right? But say, like, all of a sudden AI makes any drug that you design 100% safe and 100% effective 100% of the time, this would greatly reduce the need for these large-scale trials and things. It might, in this utopian future, you might actually see the biotechnology even become personalized. And that's very much going to benefit the individual patient. So, for example, if you have some very specific tumor with a very specific composition of mutations, right now, if you go into the healthcare system is very much not suited for this. You get like the same standard of care across the board, and a lot of people just die, even though in theory, you can treat the tumor if you had a bunch of information, if you did all the right sequencing, if you had a team of specialized doctors that can match the right therapies to the right tumors. But because that's difficult to scale, right? But if AI makes this process 1,000 times cheaper and safer, then maybe then it becomes more democratized. You might have individual drugs for individual patients, and in this setting, it actually very much benefits the patient, even at the expense of the big monopolistic pharma company. And so I do think there's definitely room to commercialize these technologies. There's room to definitely make some kind of profit on the technologies. I'm not totally anti-capitalistic or anti-entrepreneurial, and these are very much ways of helping disseminate the technology to people. But also, I think, on balance, the AI tools are great for democratization of biology and should hopefully contribute to positive outcomes for patients. So ethically, I think if we also, again, handle it in the right ways, we make sure that the right models are open, we make sure that once the technology does reach a capability advance, it does reach the broader public correctly. If we do all these things well, then I think the technology should also be ethical in the commercial dimension too.
Great. Thanks. I'll shoot for just 50%. Effective. That's a good start in that sense. So
a coin flip or something.
Right. Questions from the audience, whether things we covered or honestly things I didn't manage to cover. Yeah. And I'll repeat it backwards back so that folks online and otherwise can hear.
So are you adding phenotypic information to your training sets? That's one question. And related with that, once you have all the algorithm is trained, what type of questions are you asking? Are you asking, look for a resistant system or whatever? So what type of, how do you ask those questions, and what are the questions?
Yeah, so the two questions are you adding phenotypic information into your models? And then once that model's constructed, what next? What kind of questions are framing your investigations that are being used for it?
Yeah, it's a good question. And so the thing about the models as they're trained, it's actually quite it seems simple that the models are not trained with any explicit phenotypic information. They're just trained on raw DNA sequences. So they just predict the next DNA sequence in the next DNA base pair in a sequence. In doing so, though, the model internally gets a lot of phenotypic information within the model itself because evolution acts on phenotypes, and those phenotypic outcomes get written back into the actual DNA sequence through natural selection. So one trick we often use is, does the model think that a given DNA sequence has high likelihood or looks good? Or does a model think that a DNA sequence doesn't look good, it has low likelihood of occurring? The reason why this is useful is because the model has seen a lot of DNA from living organisms. So it knows like which mutations are compatible with life, so to speak. So if the model actually, and we can show this, if the model sees a mutation and thinks, oh, I really don't think this mutation is meant to be there, I'm going to give it really low likelihood, those mutations are, this is a very strong predictor of whether those mutations cause disease. So actually the model in many cases has state-of-the-art accuracy in being able to predict which mutations are completely benign or don't cause anything, but which mutations lead to disease. And this is a problem. This is actually a quite important problem. All of us actually have some changes in our DNA sequences. Many of these changes are probably completely benign, but in rare cases, they do cause disease. So the model, this is one type of question we can ask with the model. We can have the model identify these mutations that are incompatible with life. And maybe then the clinician can go and see, oh, this mutation is occurring in this protein, and this protein is known to have this function and connection to phenotype. So it's one way of diagnosing maybe a genetic cause for a disease. Another thing that we do with the model is, so I said the model is kind of like a black box. It's actually not. The neural network does sit on a computer, and you can open it up and peer into all the neurons in the neural network. It's very complicated. It's this dense web of neurons, but actually you can try to interpret the neural network. And a lot of these techniques are actually inspired by neuroscience. So people trying to study an individual neuron in your brain that activates when it sees a picture of Donald Trump or something. You can actually do the same thing for these artificial neural networks and peer into the neurons inside the neural network. And when we do that, we can actually see that different neurons inside our neural network will activate when it sees certain biological concepts, like that are very related to phenotypes. So certain parts of the neural network will activate when it sees a protein coding gene, or different parts of the neural network will only activate when it sees an input related to immunity. And so we can actually use these interpretability techniques to also connect what the model has learned to some kind of downstream phenotype. So we try to interpret which regions of the model produce different neurons in this neural network, different activation patterns in the neurons in the neural network. Then we connect those to downstream phenotypes, and then we have a great predictor of which parts of a genome sequence correspond to some biological phenotype. So already in a model that's only trained on genotypes or DNA sequences alone, we can extract a good amount of phenotypic information, but we also agree that in future versions of this model, what we want to do is also find ways to explicitly encode phenotypic information directly into the model. And then I think it will also, given that the model is already doing really well with zero phenotypic information at understanding high-level biological concepts, if we add phenotypic information to the model, it should improve its performance even more.
Other questions?
So it seems like right now in drug discovery development, a lot of the applications of AI are at the very beginning, right? Finding a new compound or a new biologic. But then the bottleneck right now is that FDA approval routes still require traditional clinical trials, which still cost a considerable amount of money, still take a very long time. So are we just going to reach a point where all these companies suddenly have increased tenfold or 100-fold or 1,000-fold the number of clinical trials we need to run tomorrow without any of the changes in the next part of the pipeline? So just what do you think we could do to help? Where do you think AI fits into that?
Yeah, the question, and I'll shorten it just so we can get the answer, is that there's a bottleneck ultimately between the capacity of AI to potentially generate new new lead compounds or new foundational elements of a medicine that to actually be applied need clinical trials, need perhaps multiple levels of clinical trials for some very good reasons. But is it possible that we're just going to have a backlog, for lack of a better term, of incredible, possibly compelling, possibly really useful compounds that we can't get to? And how do we address that, or should we address that? Or is that just inherent to the system?
It's definitely going to be a big bottleneck, no doubt about it. And I definitely think it will take several years, hopefully in the low numbers of decades, to clear the bottleneck. But and this is already going a little bit outside of my lab's day-to-day research, but here's how I work with a lot of companies that are also trying to get things through FDA approval. So from this experience, here's how I kind of see the progression of things going. Yes, I think in the next maybe in the 5-year timeline, we'll have this kind of huge amount of AI designed molecules trying to make it through the clinical pipeline. Where the AI tools can help in the near term is I think these large language models can help the clinical trial process become more efficient. One of the big problems is in trying to enroll patients or something, or trying to match the right patients to the right trial. These language models are really great at scouring the internet for and doing research on people and doing this. So I think that part of the clinical trial process is could benefit from the large language models. The scientific models, I think, hopefully where they can make a big impact in this first wave of AI generated molecules, and this will definitely take some time too, is hopefully there will be some generation of AI designed molecules that when they enter the clinic and go through the traditional clinical trials process, the success rate will go up from 10% to say 50% or 60%, right? And then the success rate, and this is the success rate maybe through phase 3 or like efficacy, or the success rate through phase 1, or the safety part becomes 100%. And as people begin to trust AI designed drugs better, and this is maybe on the decade or 2 decades long timescale, then maybe if you have this perfect AI predictor of safety or toxicity, then maybe the phase 1 process could actually be automated by an AI system or greatly reduced, right? And then it just becomes phase 3. So then you've like cut out a third of the clinical trial process. And then if you're in this utopian, or if you're in this setting where the AI models have 90% efficacy or something, then assuming that the clinical trial process, there's no innovation in the regulatory domain, there's no innovation in like in any of the human processes, and drugs take 5 to 10 years to make it to market anyway, you can still get a benefit if the AI models can do the target discovery, can do the synthesis of biological information, and can generate the actual molecules themselves to target the targets that are generated or that are identified by the AI with like very good specificity and developability, so that you can get 90% success rates through the clinical trial process, assuming nothing changes in the regulatory domain. You still have to, either you run the same amount of trials and get 10x the number of drugs through, or you cut down the cost of R&D or the cost of running these clinical trials by 10x or something like that. Both are good outcomes. So it will make an impact. I do think there's a lot of smart people working on, improving the clinical trials process, the regulatory process with AI. On our side, we're also doing our best to try to improve the actual the actual design of the therapeutic itself, or in designing the therapeutic to cover a wider range of diseases. So I do think it is a bottleneck. There's a lot of smart people working on it, and there should be some improvement. There's multiple ways to improve it, even if the clinical trial process itself doesn't change. There's no change in the FDA or something. Though the FDA is actually recently been more open to AI models, definitely of safety and things like this. So I definitely think even if that were frozen, if you increase the success rate from 10%, even at 20% or 30%, that's like threefold more drugs entering the market and saving lives and stuff. That'll still be a good impact.
All right. Yeah. So you're building this large language model to hopefully build, or you have built these pages. So in theory, could you use it as a very high level? Because you talk about how there's no phenotypical awareness. Could you easily construct it as a very strong annotation tool, given that there's a lack of good annotation, at least for a lot of those NCBI pipelines for those genome submissions? And would that help your tool produce better, medical solutions?
Yeah, the connection would be is, can you take this tool and because of the structure, because of what you can do with it, turn into a strong annotation tool that would have benefits in that pipeline that we've been talking about?
Yes, we are very excited by this direction. And to add some color, and you probably know this, but for other people listening, if you do the thing that I described where you go into the Amazon or you sequence the San Diego wastewater even and look at the DNA in the sewage, you'll often find DNA sequences where if you run traditional tools for telling you what these sequences correspond to, you'll find nothing, or they won't tell you anything. There's large gaps in the genome that are completely opaque to the existing tools, or the existing tools are probably giving predictions of what these DNA sequences are that are just wrong. So we actually think that these models have internalized a good deal about patterns in biology in a way that's completely label-free. So we don't bias the model towards any human knowledge. Actually, they need to learn these biological concepts purely autonomously just by predicting the next base pair in a sequence. And so we've already had very exciting work, hopefully that should come out soon, on improving the ability to kind of light up or discover these dark matter regions of the genome. And we're excited by this direction. If you think about most of the biotechnological tools that have revolutionized molecular biology over the past, century or something, they were all mostly discovered from microbes, like CRISPR-Cas systems, for genome editing, or Taq polymerase for doing PCR, things like this. There's a vast trove of discoveries in nature that escape our current tools that I think these language models can now access. And it's quite fascinating. We apply these models to these sequences. We have these ways of visualizing the annotations predicted by the model, and I liken it to there's this telescope, the James Webb Space Telescope, that has this really high resolution, and there's this famous image where Hubble imaged this patch of sky and it's completely dark because there's not sufficient resolution or something in the image, and then the James Webb Telescope imaged the same part of the sky but with better technology and just saw that it was full of actually galaxies. We're actually seeing the same thing when we look at our language model, what it thinks is going on in these sequences. You run it through some traditional tools, many of which were developed in the 90s or early 2000s, and you see nothing in this stretch of DNA, and then you put it through the neural network, and all of a sudden, a lot of these DNA sequences actually make sense. So we actually think there might be equal utility in using these molecules, in using these models for biological discovery in addition to design.
Great. We got time for another question, if you like. Yeah, wrap us up.
For I guess the generated phases 16 to 18 ones that you or that were made, I guess you mentioned that they were very similar to the ones that were found in nature. How I guess for the genes in those ones, how different was it from the known genes that were in the E. coli phage, and was it coming up with completely new proteins that were and using those as in the phage, or was it just mix and matching the different components from the phages and using that to kind of make the new phage? for generated phage.
Yeah, so the 16 or 18 successful phages that were novel and different, how or why were they different, and in what regard to other kind of E. coli phages?
Yeah. So it is a good question. So we definitely know the model is sometimes capable of generating genes that look nothing like things in nature that are functional. We've mainly shown this for genes that have lots of diversity in nature, things like anti-CRISPRs or toxin antitoxin systems, familiar with these. For the generated phages, we do actually see evidence of the model actually still being quite creative in its designs. So the one system, or one phage in particular that I like to talk about, is there's this DNA packaging protein in the phage, the J gene in 5X174. And one of the AI designs actually used a version of the J gene from a completely evolutionarily distant phage that's a lot shorter than the J gene that's in the original wild type 5X174 phage that we were trying to diversify. And people have actually spent the last 40 years taking that J gene from that evolutionarily distant phage, putting it into the genome of 5X174 and seeing that phage is actually not viable. So it's a lethal swap. One actual interesting thing that the model did is it was actually able to take that evolutionarily distant J gene, put it into the genome of 5X174, and then make a bunch of compensatory point mutations in the genetic context that then made the phage viable. And this was so interesting because the J gene is actually part of the structure of the phage and the capsid. So we actually then put the phages under a cryo-electron microscope and got an image of what this looked like. And we actually can confirm that the phage is using a much smaller protein in its capsid for the DNA packaging protein than in the native 5X174. So here you have an example of the model being able to do this kind of gene swap that human designers weren't able to do, that evolution actually wasn't able to do, and then make single point mutations in the genetic background that enabled the swap to happen, and it resulted in this very interesting structural change. So yeah, I think it's a mixture. So we definitely see genes that are diversified. We see genes that are recombined in interesting ways, where the model is also being able to design the rest of the phage to compensate for these gene swaps. And yeah, so there's a lot of diversity in these phages. That this is just one example that we decided to go down because it probably had some interesting structural change. We see like regulatory elements in the phage, for example, that are completely scrambled. They look nothing like anything in nature. If you delete these regulatory elements, the phage also dies. So the model is clearly also capable not just of diversifying the protein coding genes, but also the non-coding genome of the phage too. There's lots we could actually study in the AI-generated phages that we didn't just to narrow the scope of the paper. But it is interesting. There are lots of interesting changes in these phages. And we've talked to a lot of phage biologists that also find some of these genetic changes interesting. So lots of room for future work, definitely.
Yeah, and I would say I don't want you're really in some ways understated, so I'm going to be the overstated guy. I will be the clickbait headline. And let me just repeat back to you what you said though, is that for 40 years, scientists have tried repeatedly over and over again, perhaps over a course of an entire biologist's career, to move a particular segment of a genome somewhere, and it continued to fail. And a model, what's the run length on that model time?
A sampling run at our scale probably could run overnight or something. Yeah.
Okay, overnight.
It's longer to actually test the genomes than it is for the model to design
overnight,
things. Yeah.
Discovers not only a place to place that particular element of the genome, and then uncovers compensatory secondary changes that need to be made to make the viable. That is a heck of a thing to wake up to in the morning overnight.
Yeah.
And
it's pretty, and also evolution tried too. Like anything close to 5X174, that J gene is not there.
So I don't know if you're going to leave this feeling really excited, incredibly nervous, possibly switching to HVAC, anything, please don't. And actually, I hope you do, though, leave with appreciation as I do for Brian coming down to visit, for the work you're doing. I'll follow it. And then when you do get that Nobel, we'll say, remember when he showed up down in the library at SDSU. So come back anytime, Brian. Thanks so
This
much.
is really fun. Thanks for having me.
Yeah.